right, good afternoon, everybody. And thanks for being here, for attending Thermo Fisher educational webinar for today. We have two interesting lineup from speakers, Dr. Saiju from Millen Veteran Program and Michael from McMaster, talking about their experiences using genotyping arrays. We'll have questions at the end of the talk, so feel free to raise hand and ask your questions. And with that, I'd like to invite Dr. Saiju.
Thank you, Anu. Hello, everyone. Thank you for coming, and thank you for giving me the opportunity to talk here. So I'm going to talk a little bit about our experience with the genotyping array for the program, MVP. So most of you probably already know MVP is a national program, research program started by the VA. The idea is to sort of advance precision health by learning how genes, lifestyle, and military exposure or exposure, military experience and exposure that affect health and illness. So every participant, every veteran is a volunteer who signs a consent form for using their clinical data as well as biosample. They give I'll get into it a little bit more detail. They give a sample of blood at the time of recruitment. The VA is building the MVP cohort to curate the health and genetic data, providing it to researchers in a secure computing platform. Joining MVP is easy. There's a couple of ways. There are recruitment sites all over the country at VAs, as well as MVP Online, where veterans can go and ask to join MVP. The recruitment basically involves them completing a consent form, providing a tube of blood, EDTA blood, 10 ml, and then completing surveys. So at the time of recruitment, they complete one short survey, and then after that, we actually send them a much longer survey. So there are two MVP surveys that the veterans complete. And we merge that data with the clinical data that is available during their healthcare in the VA. To talk a little bit about the sample collection and processing. So as you can imagine, MVP is kind of all comers. All veterans are eligible to be recruited into MVP, meaning that the only requirement is that they should have used the VA healthcare system for their health care at least 6 months. Other than that, there is no disease or no specific limitations on who can join MVP. As I said earlier, we collect one tube of blood, which is shipped overnight on ice packs to our central lab. Most of the blood arrives between 24 to 48 hours. So there is uniformity. Like the samples are collected all over the country, but most of the blood reaches the central lab within the 24 to 48 hours, except when there is not the snow that you're seeing outside, but much larger snow. Sometimes it gets delayed. We accession all the samples into a LIMS system, and the LIMS system actually sort of follows everything that is done to the sample. So we have sort of a lot of metadata associated with the samples that we can then use to do the QA/QC at the end. We generally separate the plasma and the Buffy coat, and then the central lab generates DNA from the Buffy coat and then sends it out to third-party vendors for doing the molecular assays. Towards the end of the last year, we hit the 1 million veteran of the MVP having recruited, and we are still continuing going beyond the 1 million. And essentially, we have become the largest genomic data that is associated with a healthcare system where there is clinical data that is associated, longitudinal clinical data that is associated with these samples. And I'll get into a little bit more. It's a very diverse sample. So, for example, the folks with African American ancestry is one of the largest already, more than 150K. The recruitment started in 2011, and it is still continuing. Samples are collected almost daily and shipped out, and we send it for assays on a daily basis. And the data we clean and curate and release in sort of cohorts, so to speak. The last release, release 4, has about 658,000 samples. This is a sort of brief demographics. In the first 1 million veterans that were recruited, it's male heavy, as you can imagine. 91% is male, about 8% female. It's diverse. There's about 8.2% Hispanics and about 17% African American. It's an older population, so the median age is about 67. And again, there's some information here that basically says that even for example the Pacific Islanders, we have the largest Pacific Islanders group, more than 1,000. So one of the things that we do with every sample that comes in is that we have a custom MVP array that was developed almost 10 years ago now, 2014. And then we genotype every sample that comes through. So this is just to show you an example of the QA/QC. So we have the 658,000 participant data that we have released on 668 loci. And this is just to show you an example where the missingness is on the left and the discordance of duplicate samples is on the right. And as you can see, in the higher allele frequency samples, there's very low missingness, and the discordance is also very low. There is some missingness. The missingness increases as the allele frequency gets lower, and the discordance also increases a little bit. This is just, I put it in there just to show the ancestral diversity of the samples. This is PCA1 and 2, projected MVP samples in gray, projected onto the 1,000 genome. As you can see, there is a lot of admixed samples in MVP. This is an MVP. So not only do we do the genotyping, but we also do the imputation. Initially, we did it with 1,000 genome, and then 1,000 genome with the African reference population, and recently we have done it with the top med. And as you can see, in most of the, R square of 0.7, in most of the allele frequency bucket, we get pretty good imputation. It varies within the ancestral population between Europeans all the way to African, Hispanic, and Asian. So one of the projects before I get into that, so most of the data that we release is either the genotyping or the imputed genotypes. And that has been used for most of the research. More than 350 publications have come out in the last 5 or 6 years using this data. Multiple releases, from release 3 onwards, I believe, people have started publishing data. One of the things so these are good for population level analysis. One of the things that we have been interested in is sort of returning the results, anything that is actionable, for example. So starting in 2021, we decided to do a pilot study to return, to find people with actionable, veterans with actionable information, and then return the results, and in that process, doing a clinical test to validate the genotyping, the research genotyping that is done in MVP. So the whole idea was to figure out the process as well as the validity or evaluating the validity of returning these results. So this is this first project, the ROAR project, essentially returning FH results to FH patients. as you can imagine, the MVP array, the custom array that we made, has a lot of clinically relevant rare markers. And these rare markers are very challenging to genotype, as most of you probably already know. Additionally, in the case of MVP, the samples we started collecting the samples in 2011, and we are still continuing to collect. So samples are being collected over a long period of time. Samples are being assayed over a long period of time because we started doing the genotyping back in 2014-15, and we continue to do these assays. So it is very important for us, for MVP to be a single cohort. It is very important for us to do the QA/QC in a way that it is still the variability between the batches is minimized or is controlled for. So as we started doing thinking of doing the return of result project, one of the issues that we ran into is that these clinically relevant variants are very rare, and we have seen there are issues with genotyping these rare variants. Published recently, last year, I believe. And we tried using that to see if we can sort of decrease the false positive rates in individual samples, because at the population level, with the large number that we have, the variability in the genotyping can be accounted for statistically. But when you want to return individual results, we want to have accurate results per person. So this is the result of the experiment that we did. As you can imagine, the biggest response that we see by using the decrease in the false positive rate, for example, from 65 to 32, which is about 51%, happens at the lowest allele frequency. The effect of this algorithm sort of tapers out as we get into more common alleles, as expected. Essentially meaning that the common alleles are being genotyped more accurately to begin with. So one of the practical use of this is that in our the FH return of result project that we saw, before we started using this algorithm, there were issues where some of these rare variants, the ALT gene variant, for example, we were having errors in the sense that if we find somebody in MVP that has that allele, and when we do clinical validation, they don't seem to have it. So once we started using these, and an example is given here. Three of these patients, for example, was turned into no calls. So the reduction in false positive rate using this algorithm, the rare het algorithm. And since we started using it, almost 57 patients that we have done since then have all their MVP genotype has been validated in the clinical tests. Another sort of area where we have used this is precision pharmacogenomics. So VA has sort of initiated a large-scale precision pharmacogenomics where we are sort of doing pharmacogenetics, especially in the oncology setting. So as of now, more than 40,000 clinical tests has already been done. So we did a little experiment where we compared the star allele calling in from the clinical test to the MVP star allele callings. So the MVP, the custom array that we have, does not have all the star alleles to make all the all the haplotypes to make the star allele call and calling for all the relevant genes, but the ones that we could, we kind of compared. So this is basically what we did. So for the MVP array, we either run it through a PGX caller and then use the rare het algorithm to get sort of the confidence level of the low frequency allele to make the haplotypes and make the final star allele calls. And as I said earlier, some of these, MVP chip does not have all the haplotypes to make all the star allele calls. So So this is the results from that. We find pretty high concordance for some of these genes on the left here, where very small discordance, almost 97% calls are concordant. These rare calls are concordant with the clinical testing that we have done compared to MVP. So in short, basically the MVP genotyping array has sort of generated very good quality and consistent data at the biobank level over time. I want to sort of emphasize the over time part of it. It's almost 15 years and continuing in terms of sample collection, and 10 years and continuing in terms of the actual assay. And this rare adjusting genotyping has really helped us, especially in the sort of the individual level genotype calling, which is bodes very well for us to sort of think in terms of other return of results projects. With that, I want to acknowledge the veterans, of course, for making this whole thing possible. Our group at CDAX in Boston, the Thermo Fisher team, Jason, who is the PI of the MVP-ROAR project, and the VA Office of Research and Development. That's it.
Hi, everyone. So thank you for joining us for lunch today. It's my pleasure to be presenting one of the bioinformatics pipelines I developed to essentially leverage SNP genotyping arrays for an alternative motive in inferring a mitochondrial biomarker called mitochondrial DNA copy number. And here are my disclosures. All statements and opinions I express are not endorsed by Thermo Fisher, and they are sponsoring this talk. So just a little bit of a background. So I am from the Great White North in Hamilton, Ontario, Canada. And on behalf of all Canadians, I apologize for this terrible weather that we've brought to you. Yes, it's truly terrible. Though a relatively mild, Canadian winter, of course. So I come from the CRLB GML lab, and you might be wondering to yourself, why the clunky name? Well, we're actually an amalgamation of two labs. One was a biobank established in 1987, and the second was a more recent lab established in the early 2000s by Dr. Guillaume Perret, which strives to be a molecular epidemiology and precision medicine research lab. Notably, we are one of Canada's largest biobanks, so we have over 70 liquid nitrogen tanks that each house over 60,000 aliquots. While we are primarily a research lab, we do strive for excellence in quality assurance and management, and so we've attained this accreditation that's common to medical laboratory diagnostic labs. And our mission is really to support molecular epidemiology research, not only locally or nationally, but also on a global scale. And as Canadians, we have a reputation for being pretty friendly, and I'm happy to say that we're really privileged to be able to establish global research partnerships. And our biobank is not only ancestrally diverse, but it's regionally diverse as well, as we have samples from over 84 countries outside of Canada, including the US, and as far as Zimbabwe, Bangladesh, you kind of name it. Our biobank includes over 4 million specimens, most of which is blood, but we also do collect urine and other solid tissues as well. And so actually 3/4 of our biobank is collected from participants from outside of Canada. So a fairly globally representative biobank, I'm proud to say. In terms of downstream molecular testing, we support a lot of different types of testing, including clinical assays, genome-wide genotyping, and this is one of our kind of staples. We also do a lot of next-generation sequencing, proteomics testing, and methylation profiling. So really, we support this end-to-end research pipeline for global investigators, ranging from specimen collection to nucleic acid extraction to multi-omic testing, all the way down to bioinformatics, which is the sector in our lab that I spearhead. One of the missions of bioinformatics, as I'm sure many of you know in the audience, is how do we approach data sets differently, and how can we squeeze more insights out of existing data sets? So here is just a smattering of different methods, statistical genetics methods we've developed in our lab, which are available on our GitHub page. Today, I'm going to talk about a method called AutoMitoC, which uses array intensities to infer mitochondrial DNA copy number. So just a quick outline, what are the key questions that I will be covering in this talk? Firstly, what is mitochondrial DNA copy number? What are the various ways we can measure it? Is there a genetic contribution? And also, which diseases are associated with mitochondrial DNA copy number at the observational and genetic level? So full disclaimer, I am not a mitochondrial expert or biologist by any means. I am more of an enthusiast who fell in love with mitochondrial biology during my PhD thesis, which was centered around identification of prognostic stroke biomarkers. And stroke, for those of you in the audience who are unaware, refers to an acute neurological deficit lasting more than 24 hours, commonly caused by clots that block the arteries perfusing the brain. And mitochondria are inextricably linked with stroke pathophysiology, oxygen-deprived brain tissue. It eventually leads to mitochondrial deterioration, which initiates the apoptotic cascade and the death of neurons. And what was really exciting to me was some seminal work published by Hayakawa et al. In Nature, describing essentially a protective mechanism in which mitochondria are involved in resisting or making more resilient cells to temporary bouts of ischemia or oxygen deprivation. And if we think back to our high school biology class, the common analogy that's used for mitochondria is these power plants of the cell, which paints a very static picture. But in contrast, lots of publications beyond Hayakawa et al. Also show that a better analogy is that mitochondria are more dynamic, and they're actually these portable double or triple A batteries that can be swapped between cells. And this is what we see in this paper, which shows that neighboring astrocytes to oxygen-deprived neurons can actually extrude their own functional respiring mitochondria. These can be uptaken by these suffocating neurons and help these neurons survive. So this is what really motivated me to study mitochondrial biomarkers at scale. And the question was, how do we do this in large-scale epidemiological studies like these UK biobanks, FinGens, and all of us type studies. So what is mitochondrial DNA copy number? As we all know, the mitochondria genome, because it is descended from ancient alpha proteobacteria, have their own unique set of DNA. And basically, this gives us a way to quantify mitochondria in a very inexpensive way, as long as we're able to get a signal from mitochondrial DNA molecules. And more specifically, mitochondrial DNA copy number is this ratio between mitochondrial genome abundance divided by nuclear DNA signal. And the nuclear DNA copy number at in the denominator here essentially is a way for us to normalize for cell counts. So what does mitochondrial DNA copy number represent? Well, it represents the intracellular concentration, really, of mitochondrial genomes. And of course, this varies across tissue types, with metabolically more demanding tissues like the heart or brain having higher mitochondrial genome abundance relative to other tissues like our blood cells. Though, blood, because of its accessibility, is the most commonly studied milieu in which we extract mitochondrial DNA copy number information from. And in general, the literature has described that lower mitochondrial DNA copy number is associated with increased risk of diseases, and reflecting some of these processes down here listed. So my question was, could low blood mitochondrial DNA copy number be a prognostic biomarker for stroke victims, predicting, worse disability, dependence, and mortality after a stroke? So how do we measure mitochondrial DNA copy number? Essentially, as long as we have this duplex signal from a mitochondrial genetic segment and a nuclear encoded segment, then we can derive this ratio. And we can do this in a targeted manner, but I think it's even more interesting to leverage untargeted or more untargeted genetic testing modalities like SNP genotyping arrays, whole exome sequencing, and whole genome sequencing. And so I'll be focusing on the deriving mitochondrial DNA copy number estimates from SNP genotyping arrays. And just a little bit about the intuition behind array-based estimation. So typically when we're genotyping, and here is an example of a genotyping cluster intensity scatter plot for a mitochondrial variant. And we have to remember that mitochondria are exclusively inherited through our mothers, so we don't expect 3 clusters here. We don't expect a heterozygous cluster. And the contrast in conventional genotyping, the contrast between allele-specific signal intensities, is what tells us the genotypic variation. For mitochondrial DNA copy number, what we're doing is actually ignoring these 2 clusters and looking at variation within allele-specific intensities. And the idea is that irrespective of the allele, the signal intensity, if we're looking at a mitochondrial probe, should be proportional to the number of mitochondrial copies. So in this plot here, we're showing, at the extremes of the y and x axes, these individuals should have relatively higher abundance of mitochondrial genomes, whereas here, towards the origin of the scatter plot, it would presumably indicate individuals with relatively lower abundance. So here is the workflow for AutoMitoC. I have to acknowledge that, as always, we stand on the shoulder of giants, and essentially much of this work is inspired by pioneering a pipeline developed by John Lane and Dan Arking's group called the Mito pipeline. What we did was essentially we tweaked this pipeline to make it more ethnically robust so that we could apply it in our ethnically and regionally diverse cohorts. So the 3 key features or distinguishing features of our pipeline is that for the denominator, for the autosomal signal, we actually take the monomorphic probe signal intensities. As people say, one person's trash is another person's treasure. And while we often exclude these monomorphic probe sites in GWAS analysis, this actually provides a very stable readout that is ancestrally, stable, whereas with common variants, the allele frequencies will tend to vary substantially across different individuals of ancestry. So we found that this worked quite well in our ethnically diverse cohorts. The second feature is that we empirically detect any cross-hybridization of probes that may be annotated to the mitochondrial genome, but actually there's evidence of it kind of co-localizing with a segment of the autosome or a sex chromosome. And again, if we think about this ratio, especially if we count an autosomal signal intensity into that numerator, which is the mitochondrial signal intensity, this can create or add more noise to this mitochondrial DNA copy number estimate. And then lastly, we use principal component analysis to extract the mitochondrial signal. Okay, so how well does this actually work in practice? So in a study of about 6,000 individuals within the interstroke study specifically, this was an ethnically diverse sample. We did a precision medicine research array genotyping covering close to 800,000 variants. We also did a duplex qPCR as kind of the gold standard reference point, and we did see a pretty good correlation between these estimates and superseding kind of previous estimates. A key point here is the ethnic robustness of the estimates as well. So we showed pretty good agreement, again, between the array-based estimates and the qPCR-based estimates across Europeans, Hispanic individuals, continental Africans, Southeast Asians, South Asians, as well as other ethnicities. Now, do genetic factors play a role? Well, from previous studies, we know that about 10% of the variance in blood mitochondrial DNA copy number is explained by genetic factors, i.e. the heritability is about 10 to 15%, I would say. So what we did is to identify the specific genetic loci associated with mitochondrial DNA copy number, we took the UK Biobank and did a GWAS in about 400,000 individuals, and this was a cross-ancestry meta-analysis. And I'd just like to stop and reflect at this point here, saying it's pretty remarkable that with any genome-wide genotyping array, we can derive this mitochondrial biomarker, but also simultaneously understand the genetic determinants of it from the same lab test, really. And so what did we find? Well, we found a lot of loci. So we identified about 70 common variants associated with variation in blood mitochondrial DNA copy number levels. These explain about 1.5% of the variance in blood mtDNA levels, and so accounted for about 10% of the estimated heritability. So there's still a lot of missing heritability to be accounted for. What was striking is that these common genetic loci seem to overlap the Mendelian genes known to be responsible for this extreme deficiency of mitochondrial DNA copy number, characterized by about a 60 to 80% lower level compared to the average, called mtDNA depletion syndromes. And we were actually able to recover more than half of these known genes and found basically common polymorphisms that play a role in explaining more population level variation between people. And here, one great thing about the mitochondrial field is that it's quite extensive and advanced, and we have a lot of knowledge about the regulators of mitochondrial DNA. And I just wanted to highlight here that kind of the bona fide regulators of mtDNA involved in kind of the central mtDNA dogma were identified among our GWAS loci, including TFAM, which is this DNA binding protein that incurs this 180-degree bend, allowing mtDNA to condense, much like our nuclear genome condenses around our histones. And then we also identified many enzymes involved in the mtDNA replication fork. Beyond the mitochondrial central dogma, we identify genes implicated in more functional mitochondrial processes like respiration, metabolism, and homeostasis, as well as dynamics like mitochondrial biogenesis, mitophagy, fission, fusion, and morphology as well. So what diseases are associated with mitochondrial DNA copy number? This was a phenome-wide association study done for incident fee codes within the UK Biobank, and we can see that there are a lot of different diseases across many different systems involved here. Next, we ask the question, are some of these diseases mediated by low mitochondrial DNA copy number? And essentially, inspired by our finding that there is this overlap in the genetic spectrum between common and rare mtDNA depletion disorders, could a common genetic predisposition to lower mitochondrial DNA copy number kind of lead to similar features that we see in these individuals with extreme mtDNA depletion disorders? And so this is a targeted kind of Mendelian randomization analysis, and what we found was that genetically predicted lower levels of mitochondrial DNA copy number were associated with a doubling of dementia risk. And this result has also been replicated by Shay Andrews from UCS. So this was a little bit of a detour. So going back to the initial question of, is low mitochondrial DNA copy number a good prognostic biomarker for stroke? And indeed, we did find this. So just to orient everybody a little bit, what we did was we split individuals based on their mtDNA-CN levels into quartiles from lowest to highest. And then we looked at 3 outcomes, ordinal modified Rankin scale. So this is a scale ranging from 0 to 6, which reflects functional dependence of an individual. So 0 indicates no symptoms. 3 indicates an individual who would depend on another to execute some of their daily activities of living. And then 6 is the most extreme, indicating death. And then functional outcome is a dichotomized version of this ordinal scale, comparing 0 to 2 versus 3 to 6. And then we have mortality as well as a hard endpoint. Redder shades indicate more favorable outcomes, and bluer shades indicate less favorable outcomes. And what we can see is that as mitochondrial DNA copy number quartile decreases, there's this shift towards worse outcomes. And the effect size is comparable to that of established prognosticators for stroke, like ApoE4, as well as older age. So that was an observational study that we conducted. To follow up on this, we asked the question, could restoring mitochondrial DNA copy number be a potential therapeutic target? And so we did a Mendelian randomization analysis, and indeed we see a similar effect whereby genetically lower mitochondrial DNA copy number was associated with an increased risk of higher ordinal MRS, as well as poor functional outcome status. Important to note here is that cell composition or blood cell proportions is a strong confounder of mitochondrial DNA copy number. So as a sensitivity analysis, we also evaluated potential causal effects of different blood cell counts on stroke prognosis. And as you can see, based on the thinner confidence intervals, we had much better instruments for these cell counts, but despite that, we did not detect a significant MR causal effect. So in summary, mitochondrial DNA copy number is a mitochondrial biomarker that is extremely accessible from different genetic testing modalities and relatively inexpensive. genetic factors do play some of a role in determining blood mitochondrial DNA copy number levels, and future studies should continue to elucidate these specific loci. Perturbed mitochondrial DNA copy number levels might be both predictive and prognostic for some diseases. And in some contexts, like we showed for stroke, it may even be a mediator. And then lastly, I just like to say that we developed this AutoMitoC pipeline to really enable accessibility to derive this mitochondrial DNA copy number in global studies and ethnically diverse populations. And this is kind of a neat, I think, add-on to existing data sets. And we'd be happy to support any interested parties in adding this to new genotyping initiatives as well. So thank you all for listening, and I'd just like to acknowledge the hard work of the entire CRLB Gmail team, especially my mentor, Dr. Guillaume Perret. Thank you.
Hi. The MVP data was very impressive, the concordance with the clinical testing. Are there plans for an updated version of the array with additional PGX markers, or to tweak the ones that are on there to increase that very impressive concordance?
Very interesting question. So we are sort of fighting two things, right? One is that we want to sort of the harmonization of the data set, right? If we want to use the same array set to see if we can have, as I said, we are still continuing to do the assay, and we are still continuing to do collect samples. So we would like to keep the same, at least, for the 600,000 plus variants, we would like to keep the same array. One of the things that we have done, so I didn't talk a little bit, I didn't talk much about it, is that the original array that we had developed imputes very well for European population, but does not impute too well for other population. So we have actually generated a second array that sort of complements the first array, so to speak, and improves the imputability of the other ancestries. So in terms of pharmacogenomics, that's one of the ideas that we have, that we could, instead of doing a completely new array, we could do an array that sort of complements what we already have. Great question, though. We think about that all the time, the longitudinality of genotyping.
Hello, thank you for the very, very interesting talks. I have two quick questions. Number one, have you guys explored at all calling mitochondrial copy number from whole exome and or whole genome sequencing? And if you have, are they, how correlated are they with genotype arrays? And number two, I think you touched on this, but can you speak a little more to the view as to what component of mitochondrial copy number is coming from DNA derived from blood cells or hematopoietic cells versus other tissues, how that might relate to seeing signal from neurological phenotypes?
Great questions. So yeah, in our eLife paper, we did publish concordance estimates with whole exome sequencing, also parallel whole exome sequencing derived in the UK Biobank. At the time, there wasn't whole genome sequencing, but the correlation was pretty good. I think it was around 0.4, 0.5, so less good than qPCR. But with whole exome sequencing, there's all sorts of biases with the target enrichment. So yeah, that's why we went ahead actually and did the qPCR as maybe a more stable level of ground truth. I think whole genome sequencing is the platinum standard. And at the time, the UK Biobank whole genomes were not available, but yeah, I mean, I would love to revisit that. And then your second question is, yeah, one that I am very interested in as well. So obviously there's extracellular vesicles floating around in circulation, which could originate from other tissue sources, and so that would not be currently captured because this is all based on buffy coat, right? So mainly white blood cells. But yeah, I do have a student that's interested in looking at the relationship kind of between intracellular and extracellular mitochondrial DNA copy number levels. And yes, so figuring out which cells that the extracellular content is coming from is not trivial. I think, I'm not sure if this is a controversial statement, but one way that we can quantify cell identity is, so this won't be a controversial statement, this first one, but one obvious way that comes to mind is DNA methylation, right? For cell deconvolution and identification, but mtDNA is not methylated, or maybe not methylated in the same way that the nuclear genome is. So we can't really leverage that, but I'd love to discuss further maybe afterwards to see how we could get at this. Yeah.
This question, great talks. This question is related to the first one. So for the array, the mitochondrial copy number calling, does the common Illumina arrays like the global diversity array and global screening array, do they have the contents for this method, or do they need custom contents, added probes to use your method?
Yeah, that's a good question. I have not directly tested on those specific arrays. What I do know is that we did some kind of internal analyses to see, how much information do we actually need from the autosome and also the mitochondrial genome. So what is the minimum number of, SNPs or probes that we need? And for the mitochondrial signal, I think we were able to get down to as little as maybe 30 mitochondrial probes. For the autosome, we were able to get down to, we only tested down to 1,000 monomorphic sites, but that was more, than enough to maintain the, correlation with qPCR. And it's also extremely fast for that reason, because we're not looking at hundreds of thousands of signal intensities. We're just looking at maybe a little over 1,000. Yeah.
Mm-hmm. I have a question with the first speaker, like the rare variants. We have been using the rare variants calling called Z-call. I don't know if you heard of that. I just want to know the method you presented. Are they similar, or how different they are? Or?
No, we have been sort of generally using the general genotyping methods, and we have just adopted that.
Okay.
Hi, so my first question is for Dr. Chong. So I was really curious about how you account for heteroplasmy, because, that's not necessarily trivial, but it's not, maybe you agree, maybe you don't, something that we can ignore.
So I do agree it's not trivial. Mm-hmm. I did have a separate project trying to look at heteroplasmy and to basically call heteroplasmy from arrays, but it was very difficult. I would say that we have done some sensitivity analyses to basically what we did was we chopped up the mitochondrial genome into tertiles and derived the numerator based on different tertiles, to make sure that we weren't, biased to any particular set of mitochondrial probes, and that it does reflect a more, general abundance. So I know that there's some sites that are more prone to heteroplasmy, so hopefully we are guarding against that. But no, it's a great question. Yeah.
Thank you. My second question is, how are you accounting for the difference in allele frequencies across your different populations? You said you have a second panel that kind of complements the first. At my institution, we only have one, and it's always kind of a juggle of which one we're going to put on. There's something like DPYD that we're very interested in. It's very rare, but it's very clinically relevant, right? So kind of how do you go through that?
So one of the things that we have done is that because of the larger population size, we have actually calculated allele frequency at the sub-ancestry level. That's one thing that we have done. And then the second thing is that essentially sort of grouping the star alleles such that we are calling the star alleles within each ancestry rather than the global population.
Makes sense.
Thank you. Thank you both for the great talk. My question regarding the Million Veteran Program. You did refer that you collected blood samples either at the recruitment site or remotely using self-collection kit. I don't know if you are referring to the capillary system or for the self-collection kit. What system did you use? Are you referring to the capillary one or how they collected themselves? And what challenges did you face with that? Did you have enough emails of blood, enough DNA and the quantity? Yeah, thank you.
So great question. So when we started the recruitment in 2011, it was, we had like roving recruitment sites at different VAs across the country, and every sample was collected by a phlebotomist on site. The remote or self-collection basically started because our recruitment came to a grinding halt with the pandemic, and that so there are, as you're mentioning, there are lots of issues associated with the quantity of the samples, the amount of DNA that you get, and especially in our population, which is older population, it's much harder to get enough by the capillary method that we have been using. So yeah, we have been not too happy with the with the self collection because we have such good quality of samples from the site collection. Yes.
This is also a question for the first speaker. So I noticed in the genes that you guys were able to predict with your data, the screen flashed by quickly. I don't know if CYP2D6 was part of that. I thought it was missing. But do you guys have any plans to make it more functional to include important genes like CYP2D6?
Correct. So as I said, like our original custom chip did not have all the, back when we designed the chip back in 2014, we didn't have all the enough information to put in all the pharmacogenomic haplotypes to call the star allele. So as I was answering that first question here, that's one of the thoughts that we have. We have to supplement one way or the other. We have to supplement what we have in our original array to do better, star allele calling.
And just a follow-up question, do you guys have plans to use that information for decision-making for individuals at any given point?
So we are thinking of it, so remember this, the original MVP array and the original data generation is not clear, is completely for research. The idea of return of result for us is that, and we chose that sort of knowingly because we wanted to do a large data set at the cheapest cost, as quickly as we could. And the idea was always that, if there is actionable variance that we discover at the population level, we can then sort of have more specific clinical test done so that it'll be more, we can one is validate the original array, number one. Number two, then we can have a more, in terms of time, we can have a more proximal test to whatever the action that needs to be created. So that's how we are thinking about it.
Thank you.
All right, thank you both. I just have a question about AutoMitoC and how you accounted for NUMTs or nuclear embedded mitochondrial regions.
Yeah, so great question. So maybe just for the broader audience, over like evolutionary history, the mitochondrial genome has become smaller and segments of the mitochondrial genome have introgressed into the nuclear genome. And so basically what this means is that there is, sequence homology. And so we could be miscounting, as you're suggesting, autosomal num-Ts as mitochondrial signal when we shouldn't be doing that. So the main way that we've guarded against this is kind of the approach that has been done by others and that I just described before, which is to make sure, or it's to do these sensitivity analyses, chopping up the mitochondrial genome into tertiles. We also do take a specific look at the probes that have high sequence homology with the num-T regions, and we exclude that from our pipeline as well. But yeah, you can't capture them all like this.
Thank you. Yeah, I was just curious how you curated the list then of the num-Ts specifically that you were trying to look for.
I cannot remember the database that
Okay.
yeah, has been.
Because
that has been
this is
used
just
for
a
this.
question we come across too. So
Yeah.
yeah.
Yeah, no, I'm happy to discuss afterwards as well.
Awesome. Thanks.
So I have a question for Dr. Saiju. So when you guys started the MVP program, what prompted you to use an array-based technology over next-generation sequencing?
Great question. So MVP sort of started off in 2010, 2011, right? And as I said earlier, our aim was to get the largest number of samples collected and assayed in the shortest amount of time and the minimum cost. And at that time, the whole genome sequencing, as you can imagine, was very expensive. We actually thought of whole genome sequencing initially, but then we switched to genotyping. But over time, we are doing the genotyping on every sample that comes through, on every participant that comes through. But our idea is to have a multi-omics, sort of a nested multi-omics. So on the same samples, depending on the cost, we are doing whole genome sequencing, methylation array, proteomics, and metabolomics on these subset of samples. So a small subset of samples have already received all these omics that I just mentioned. Our idea is to keep increasing that.
All right, guys, thank you for joining us. Round of applause for our speakers. Right, have a brilliant afternoon.